Okay, class. Sorry, I'm late. I had to take some stuff to the dump, and I just got back, and then I couldn't log into the do other things. So. But I'm here, so everybody can hear me.
Yes.
Awesome. All right, so let's get started. You have any questions?
No.
Okay. So we're getting close to the end. This is the material for the next test. So we covered chapter 10, and chapter 12 has to do with the cell cycle. Parts of this chapter we're going to skip because of time, but this is super important because this has to do with how your cells divide, how you went from a single cell to the trillions of cells that you are today. So there's a few roles of the cell cycle. So reproduction is one. And so we have to go through a cell cycle, and typically the cell cycle is drawn as a circle. You can think of it as like a pizza cut into 4 slices. And then we have G1, which is growth 1, or also gap 1. S stands for synthesis, where the DNA is copied. Remember, you only have 1 copy of your DNA, so if you're going to make a new cell, you need another copy. And then G2 is to check, make sure the DNA is copied correctly. And M is, the M phase stands for mitosis, but it also stands for meiosis. These are 2 different events. The goal of mitosis is to make identical cells, and we call these somatic cells, which are all the cells in your body except your sex cells. So we mean like sperm and egg. And then meiosis, its job is to make sex cells. And you want to make sex cells that are different, like sperm and egg, because if you didn't, then all the kids that you had with the same person would all look identical. There'd be no variation. So meiosis, we want variation. We're making sex cells and we want half the amount of DNA. And mitosis, we want them to be identical cells for all the other cells in our body, which we call somatic cells. Growth. So just a shocker here. You guys came from a sperm that fertilized an egg. Even if that happened at a test tube or naturally, it doesn't matter. And that made what we call a zygote, which is a fertilized egg. That once that egg is fertilized, it has 46 chromosomes. Each of these have 23. Remember, you got 23 from mom and 23 from dad. And we talked about that before. And then that zygote, that single cell has to grow into trillions of cells and differentiate. So some are going to make your heart cells, some will make your brain cells, your lung cells, your skin cells, and so on and so forth. So you have to go through, hundreds of thousands of cell divisions to get trillions of cells that make you what you are. And so that is what we consider growth, right? You came from a fertilized egg to a multicellular organism. And then you also need cells division to make new cells if you get a cut. So let's say you got a cut on your arm. Here's your arm. It's a terrible arm, but that's okay. Here's an arm. You got a cut on it, right? So you have a gap in that space. And those cells have to divide. You don't really see this because it's scabbed over or whatever. But underneath the scab, those cells are dividing to fill in that hole that you made from whatever you did to your arm. And so repair of tissue is really important. You have to replace damage or even cells that are dead. This could happen from getting something as simple as a sunburn or an actual burn. All right, so those are the roles of the cell cycle. The reason that cells go through this is either to do mitosis, which would involve growth and tissue repair, or meiosis, which would involve reproduction. Okay, so a lot of things have to go on in cell division. So, keep in mind that we have to replicate the DNA. This is no small feat. Remember, our DNA is 4.2 billion AGCs and Ts. To put that in perspective, that would be your textbook, like the height of the Washington Monument. So that's a massive amount of information. It has to make identical copies of each of those chromosomes, every one of the 23 that you got from mom and the 23 you got from dad. And if it screws up and the copies are different, what do we call that?
Mutation.
Yeah, and mutation leads to bad things. Most human diseases are caused by mutations. And we'll talk about how it does this in chapter 16, but it's called semi-conservative. All right, so once it's copied all this DNA, you can kind of, so DNA is long and stringy, right? It's, and you have 23 of these from your mom, and I'm not going to draw everyone out, and then 23 of your dad. So imagine that you had 46 sets of headphones in your gym bag, and you just threw them in there. So, and you shook it around or whatever, like you're walking around, just like you do in your cells. And then it comes to, for you to separate out all of those different headphones because you want to get your favorite pair. And what's going to happen when you pull out all of these 46 pairs of headphones?
They would be tangled.
Yeah, they're all going to get tangled. And if you pull them hard enough, what happens?
They'd knot and break.
Yeah, they would rip and break, and you don't want your chromosomes to break. So this is actually quite a complicated choreography to get all of these 46 sets of, and they're not headphones, they're chromosomes, right? To separate them from one another. And so the way that this is done is kind of like how you would make sure that your headphones stayed separated. You kind of wind them up individually, you know, and make them compact so that they weren't all stringy and all over the place. And so part of this cell division is to separate the chromosomes from one another, and we have to physically move them. So let's say you have 46 chromosomes in this cell. So you want to make 2 daughter cells. You're going to have to copy this DNA, and you'll end up with 92. And then when you have that, each of the new cells will have 46, right? So you got to make sure not only that these things don't get tangled up or whatever, but that this ends up with 23 exact copies of mom and 23 exact copies of dad from this. And remember, that would be the equivalent of you retyping your textbook that was the height of the Washington Monument and not making any mistakes, right? So that's not an easy feat. And then the same thing would happen with this cell too. So we want 2 cells that are exactly genetically identical to each other. And then they have to get moved. Those chromosomes have to get moved to separate places. And then once we do that, so once we have the 92 chromosomes, we have to physically cut the cell in half to make these 2 new cells. So it physically divides the cell into 2 segments. And that's basically what's going on in cell division. This is a simplified version. It's very complicated. There's whole classes you can take on cell biology that describe how this works. But we're just going to touch on the most important part. This should be a review. You guys should know what the word genome means, which is, remember, it's the total genetic instructions that make up an organism. And every cell in an organism has identical copies of its genome. Remember, you came from a single cell. So every cell that came from that, the DNA got copied. And so all of those cells have the exact, every cell in your body has the exact same DNA. Genomes can be small. Bacteria, viruses can have relatively small one. Viruses have what we in the thousands of nucleotides. Bacteria have in the millions of nucleotides, and then most eukaryotes have in the billions of nucleotides. So we're talking like, you know, thousands of orders of magnitude larger between each of these organisms. And the only way that we could manage this massive amount of information is to organize it into subunits, these chromosomes. And we've talked about chromosomes before. Remember, chroma means colored and soma means part. All right, so if we took a DNA out of one of your cells and stretched it out, it would be about 7.5 feet long. That's a lot of DNA, and we have to package that into to fit inside the nucleus of a cell that you can't even see with your naked eye. So the way that that works is the DNA is actually wrapped around, and I think I may have told you guys this kite spool story, but when we were kids, we didn't have video games, and I mean, we had Pong, but that got boring after a while. So we had to go do stuff outside, like fly kites. And one of the things me and my friends would like to do is we like to fly kites and see how high we could fly them. So we would go to the store and we'd buy spools and spools and spools and spools and spools of kite string. Like most of the money that we got from allowance, we spent on kite string and kites. And I think at one point we had like a little over 5,000 feet of kite string. And so one day, we were out flying our kites. We'd been doing that all day since the morning, just adding string to it. And then my father came home, and of course that meant it was time for dinner. So I had like 15 minutes to pull in a mile of kite string. And so what I did was I didn't have time to spool it up. So I just started pulling on the string and pull it. And then I took that whole giant ball, that gob of string, and threw it in the garage. And then, of course, my father saw that, and he was super pissed off at me. So what do you think takes up more space? That giant gob of string that I threw in the garage, or the 100 or so spools that I made it with?
The big ball.
Yeah, the big ball took up way more space. And so it's kind of the same way with DNA, right? If you want to package it, then you need to wrap it like a kite string around a spool. And so the spools are called histones. These are made out of protein, and they literally are round. They're circular, just like a kite spool, like a string spool. There are a bunch of subunits that make this up. You don't need to know that. If you took genetics, you would. And so it's not just DNA that makes up our chromosomes, but it's DNA plus these proteins that the DNA is wound around so that it'll fit inside the nucleus. And this is the DNA in blue, and in red right here are those histone proteins. So make sure you guys know what histones are, and that make sure you know that DNA plus histones makes up chromatin, and chromatin, long strands of chromatin, make chromosomes. Any questions?
No.
Okay. So if we looked at a human, and we've talked about this before, you guys know that you have 23 pairs of chromosomes, 23 that you got from mom and 23 you got from dad. And when scientists discovered these, they actually started numbering them, and they numbered them based on how long they were. So they don't look like this in the cell. What happens is that they do what we call a karyotype. So they take a cell and they squash it between two slides, and the guts of the nucleus come out, the chromosomes. So this is what it looks like. Back in the day, they would have to cut this out with like actual scissors and then glue it together on a poster board. Today, you guys know that we can cut and paste stuff relatively easy in Photoshop and things like that. And so they will line them up. So if you were going to have a baby, you might want to do a karyotype to see if it has an abnormal number of chromosomes. And so they line them up based on length. So 1 is the longest, 22 they thought was the shortest, but I'll get into that in a second. But so these are progressively shorter and shorter, which means they have less genetic information in them. And then we have sex chromosomes, which are called the X and the Y. So if you have 2 X's, you're a female, and an X and a Y, you're male. And we should have 2 chromosomes for, so we should have one number 1 that we got from mom, and we should have one number 1 that we got from dad. On here, maybe it has a gene. I'll just start over here. So on here, maybe there's a gene for eye color, and the one that you got from mom, the gene makes a blue pigment, and the one you got from dad makes a brown pigment. Right, so they're not identical. They're just identical in length, and they're identical in the genes that they carry. So this would be a gene for eye color. There's actually more than one gene for eye color, but it's just easy to do this way. All right, so does everybody understand how karyotyping works? We're just matching up the chromosomes. So if you had an extra 21, so if there was 3 of these, then the doctor would come back and tell you that your child has Downs syndrome. And there's not a lot of survivable extra chromosome conditions. There are no survivable too few chromosomal conditions. So if you only had number 115 chromosome, you would not be born. When we look at the cell cycle, remember I told you it's like a piece of pizza, and we cut it into 4. We have the G1, S, G2, and M phase, and that M is mitosis or meiosis. So interphase is the time of all this other stuff that's going on. Growth, DNA replication, checking the DNA to make sure it's copied correctly. So that's called interphase. And then the M phase, this phase right here is the mitotic phase. So this is interphase. And I think I have a drawing that was done by an artist on the next slide. And a couple slides. Okay. So you don't need to know this. What you guys should know is this form right here, the most common form that's represented is called the 30 nanometer form of chromatin. And you'll hear that again, but I just kind of, this is just to show you that when we're looking at a normal chromosome, you can't see it. So let's say that this is your number 1 chromosome from your mother or father, sorry, and this is the number 1 chromosome from your mother. So in the cell, these are, if they're not packaged real tightly, because if they were all wrapped up, it would be like taking a scroll and rolling it up. You wouldn't be able to read what the scroll says until you unroll it. And so the same thing with like headphones. If you had on the wires written, the brand or whatever, you wouldn't be able to read the brand of them until you unwrap them. And so just like that, we wouldn't be able to read the genes for, say, eye color. So we wouldn't know, we wouldn't be able to take the DNA and make RNA and make protein out of it for that blue pigment or that brown pigment. And so the chromosomes in interphase are not condensed. They're like open scrolls, so you can read them. But when we go to M phase, the mitotic phase, they have to get packaged like the headphones in the gym bag, because otherwise they'll get all tangled together and they'll tear apart when we separate them. So this is how it goes through M phase to tighten these up. So this is how it would be relaxed, like in interphase. And in M phase, it starts tightening up into these coils, and it gets packaged tighter, and then it gets looped. And in the end, it's real condensed. And the reason that this looks like an X, this would be like mom's chromosome that got divided in S phase. Remember we said we have the pizza in G1, S, G2, and M. So in this phase, S phase, this is mom. She would get a copy. Her chromosome would have to be copied, right? Because it has to go to the daughter cell. And so this would also be a brown gene. And they're held together to keep them organized by this thing called a centromere. And we'll talk about that in a second. So this is actually in M phase, and that's why it looks like an X, because we can't see the chromosomes in interphase. They're relaxed. It's only when they're condensed like this. And by the way, this is about 50,000 times shorter and fatter than an interphase chromosome. This is an M phase chromosome. All right. So what I want you to take away from this slide is that you cannot see chromosomes in interphase. You can only see them when they're condensed, and they only become condensed in mitosis. And that's after they're replicated. So that's after S phase, because we're going like a clockwise. And you should know the 30 nanometer fiber. That's the common one that people talk about, which refers to M phase. All right, so this is just showing you what we just talked about. Let's say that this is the #1 chromosome from your dad. It's going to get copied. So let's say that this had blue eyes on it. When it gets copied in S phase, it's going to get copied identically. So this would also have the gene for blue eyes on it. And then whenever we go through M phase, so when it gets copied and they're held together by that centromere, which is right here, we call these sister chromatids. You're going to need to memorize this, because this is how geneticists and scientists talk, and you just can't get away from it. It's like if you started working in a hospital and you didn't know what stat meant. That's not a common word, right? That means right away in Latin. So you know, this is just how biologists talk, and you're just going to have to learn the language. So sister chromatids are identical copies. So let's say this is the #1 chromosome. This is from dad. This is the #1 chromosome, and this is from mom. So when it gets replicated in S phase and they're held together by a centromere, these are called sister chromatids. Same thing here, right? And this could be brown. So if this was copied, this would also be brown. And this could be blue. And if this is copied, this is also blue. So these are also sister chromatids because they're genetically identical. Brown, brown, blue, blue. Now, because they have the same genes, like eye color, and they're the same number chromosome, but they're not identical, they're called homologous. So this would be like the #1 chromosome from mom and the #1 chromosome from dad. Those are homologous. So mom might have brown hair and blue eyes, and dad might have brown hair and green eyes. They can all be different on there. They don't have to be the same. But if they're sister chromatids, they're identical copies that resulted from the cell cycle S phase. And so you need to know the difference between sister chromatids that are identical copies and homologous chromosomes that are similar, like a #1 chromosome that you got from your mom and a #1 that you got from your dad. Any questions about that? So make you some flashcards and make sure you know the difference between sister chromatids and homologous chromosomes. I'm going to talk like that for the rest of the lecture. So hopefully you guys will pick it up by the end of this. All right. So once we go, so now we're not talking about the interphase. So we're not talking about G1 and S and G2. We're talking about M phase, which is in this chapter it's mitosis, and in chapter 13 we'll cover meiosis, which is making sex cells, right? Mitosis is making identical, what we call somatic cells. All right, and then after mitosis we have something called cytokinesis. So cyto is cell, kinesis is moving. Cytokinesis is basically taking the cell, cutting it into, I think I talked a little bit about this before when we were talking about the cytoskeletal components, but It's like taking a lump of clay and wrapping some dental floss around it and pulling it tight. That would get pinched in two, and you would end up with two cells. The phospholipid bilayer gets separated. So that's what cytokinesis is, cell splitting. And so we do mitosis and cytokinesis, and that produces two genetically identical cells from a single. So somatic cells, you need to know that term. I'm sure it's in the crossword puzzle. Somatic cells are all the cells in your body except sex cells, and then you have sex cells, which we call gametes. And you'll need to know that too. All right, so mitosis makes somatic cells, meiosis makes gametes, sex cells. And mitosis has 100% of the genetic information, but sex cells only get half, because when the sperm fertilizes the egg, when they come together, we have to have 46 chromosomes. If this one didn't have half the amount of DNA, if it had 46 and 46, you'd have 92. And that means you'd have four number 1 chromosomes, right? And that would kill you. Because having 3 number 21 chromosomes gives you Down syndrome. So having 4 number 1 chromosome, or any chromosome, you would never be born. You'd be a miscarriage. So it's super important that you get half the genetic information through meiosis, and that we get identical genetic information through mitosis. Any questions about that? Okay, so I've been drawing the cell cycle the whole time. Here it is. Artist drew it. It's a lot better than my drawing. G1 is growth or gap 1. So what happens is you have a cell, right? And you're going to divide it in two. So let's say I gave you a piece of Play-Doh and you divided it in two. And you not only divided it in two once, but you divided it into 10,000 times. What would be the size of the Play-Doh at the end of your 10,000 divisions? Would it be the same as that you started with?
I'm sorry, what was the question?
So let's say I gave you a piece of Play-Doh. Let's say I gave you a piece of Play-Doh and I asked you to divide that 10,000 times. In the end, each of those pieces of Play-Doh, the 10,000 ones, would they be the same size as the one you started with?
No.
They'd be a lot smaller, right?
Yeah.
So what if you wanted them to be the same size, what would you have to do?
Add more.
What is that?
Right, you'd have to add more. And so we call that growth. So the first thing that has to do is your cell has to grow. It has to roughly double in size before we cut it in half, because we want the daughter cells to be the same size as the mother cell. And then we know that we have to copy the DNA, right? So you have 46 chromosomes. We have to copy that. That gives us 92. And then when we divide them in two, we'll end up with the same number we had to begin with, which is 46. G2, or gap 2 or growth 2, is checking the DNA, right? All kinds of bad things could happen here. Let's say you laid out in the sun. Well, when you lay out in the sun, let's say you have A and G and C, and you have these two T's. These two T's get linked together by sunlight, like ultraviolet light, and that creates what we call thymine dimers. They're covalently linked. And you guys remember about covalent bonds? Can we undo them?
No.
That's right. So we have to cut that piece of DNA out. So the longer that you lay out in the sun, the more that the enzymes have to work to cut the DNA to fix it. And if you lay out in the sun too long, then it can't fix it. So there's an enzyme in G2 called P53. And P53 checks the DNA. And there's two options. you can't undo the DNA. You've already doubled the amount of DNA, so it's not survivable with the amount of DNA that it has. So you have to go through the cell cycle. Or the other option is that you can go do apoptosis. Do you guys remember what that is?
Cell suicide.
Yeah. So if you've been out in the sun long enough, your cells have to make the decision. Are they going to let those mutations go on to the two daughter cells that will also pass them on, or is the cell going to kill itself and just end the mutation line right there? And so if it can fix it, it'll go on, and if it can't, then you get a sunburn, which is basically your cells killing themselves, and then your white blood cells have to go in there and eat them to get rid of them. So that's the purpose of G2, is to either make the decision to go on to M phase or to kill itself. And so let's say that you weren't in the sun that long and it was able to fix it, you would go on to the mitotic phase or the M phase to make two new skin cells. So that's the cell cycle. Remember, interphase is all the other area besides M. So we have basically M, G1, S, and G2, and then interphase is everything. Interphase includes G1, S, and G2, and the mitotic phase we'll get into in a second. It has 5 different steps. Do you guys have any questions about this?
Nope.
Okay. Okay. So let's say you got a cut in your leg. So your cells would be what we call G0, which is rest. They wouldn't keep dividing, because let's say if your skin cells kept dividing and dividing and dividing and dividing all the time, what would you end up with?
Wouldn't that give you skin cancer?
Yeah, it would be what we call a tumor, right? And you don't want that. So your skin cells like let's say you cut yourself, they would grow together, and then once they touched each other, they would stop and they would go into rest. So they would just sit there until you cut yourself again or something happened, or you killed your cells by laying out in the sun. But most cells are going to be at rest. Like your heart isn't getting bigger. The cells may, but you're not getting new cells. Same thing with your brain, even though you might feel like it. Your liver isn't getting bigger, or there may be a problem. So most cells are at rest. Some cells are always dividing. So some cells, like the cells that line your intestines, your stomach and your small intestines and your digestive tract, those are constantly getting replaced. The ones in your lungs, they're constantly getting replaced. The cells in your immune cells, your red blood cells, those are always going through the cell cycle. They're never at rest. There are some cells that are always at rest. They never go through the cell cycle. So like the nerve of your spine, your spinal column, right? If you severed your spinal column, it wouldn't grow back together and you wouldn't walk again. Like you would be, a quadriplegic or a paraplegic, because we just don't know how to make the nerve cells come out of this resting phase. But wouldn't it be cool if you could and make it go through the cell cycle? And then people that had spinal injuries would be able to walk again. And, another thing is like, we know that if cells continue to go through the cell cycle and they never stop, those cause tumors. So wouldn't it be great if you could stop the cell cycle if you wanted to? In cancer cells, what would happen?
They would die.
Yeah, and you would cure cancer. That'd be a big deal, wouldn't it? You could make people walk and you could cure cancer if you understood how the cell cycle works. So this is a big fat deal right here. And that's why we spent some time on it. This process of copying these cells is super reliable. It only makes one mistake per 100,000 divisions. Okay, so let's break down mitosis. So there's 5 phases that your book recognizes. Some books only do 4, but your book does 5. So we're just going to stick with that. So we have prophase. That means before, like prokaryote. Pro-metaphase, it means before middle. Metaphase is middle. Ana, remember ana means up. And then telo means end. So that if you know those roots, we're going to talk about telomeres. See if you can tell me what those mean, what that word means.
Between the endings.
So what is mere? What is meros? Remember we learned about isomers.
It's the shape.
Well, it's the iso means name and meros means part. So what's a telomere?
The part in
End
between.
The end part, the end of the chromosome is the telomere. So telophase is the end of mitosis, right? And then remember we talked about anatomy. Ana means up and tomi means to cut. So anatomy means to cut up. So if you remember these roots like prokaryote, then it makes it easier to sort of know the order of these phases. All right, so we're going to go through each of these individually. And mitosis, remember this is not meiosis. We're just going to make somatic cells. We want them to be genetically identical. These are cells that are going to be your body cells. So everything but sex cells. So prophase, remember pro means before or beginning or whatever. So this is the first phase. Remember we have a nucleus. Remember that nucleus we learned about in chapter 6. And that houses our DNA. And there are these things called centromeres that hold the chromosomes together. Remember, so we know we wouldn't be able to see these chromosomes unless it was M phase. So we know we're in M phase because we can see them. They look like little worms. And then we have these things called centrioles. They're also called microtubule organizing centers because they're basically build microtubules. Remember microtubules? We learned those to be. I don't know why I can't spell the name. Microtubules. Remember we learned about those when we learned about the cytoskeleton. So microtubules are kind of like Lego blocks. You stick them together and you can make long pieces out of them. So they're just building these microtubules to come out. And the job in the end of it is they're going to attach to the chromosomes. But what's the problem here? Why can't they attach to the chromosomes? What's in the way? You guys there?
Is it membrane?
Yeah. What is that? That's the nucleus, right? So the nuclear membrane is in the way. These can't attach to the chromosomes yet. So one telltale sign of prophase is that the nucleus is still intact. And you can see that here. It's nice and round. The centrioles are made and they're building. So in green are these microtubules. And they're trying to attach, but they can't do that. So the nuclei are gone. We learned about this in RNA synthesis. Remember, that's a little dark circle in the nucleus. And that the reason is because everything is shut down. The chromosomes are condensed, just like rolling up a scroll. No DNA can be read at this time. The chromatin, remember that's DNA plus histones condenses. So we've twisted, it's kind of like twisting a rubber band. So you keep twisting and twisting it, and it's going to get shorter and fatter. And so that's what we mean by condensing the chromatin. And we do that because we don't want it to tear. We don't want the long strands of DNA to tear. And then we have these mitotic spindles, these things from these microtubule organizing centers make microtubules. Those are to pull the sister chromatids apart. Remember, the sister chromatids are the copies of, say, mom's #1 chromosome. And the centrosomes are these things. That's what these are called, centrosomes. These two little rods in here are called centrioles, and this whole thing is called the common term is microtubule organizing center, but the old school term is centrosome. These things move to opposite sides of the cell as well, because they need to set up a force. Think about it this way. Do you guys, can you imagine having a pen with a cap that's on it?
It wouldn't work.
Could you pull that cap off with one hand?
Maybe. I mean,
Yeah, no, I'd like to see it. You would send me a video. You can't. I mean, unless you like threw it really hard and you got it, you did some sort of centrifugal force to get it off. But you wouldn't be able to, and you need two forces that are pulling on different directions, right? You need to hold on to the pin, and you need to hold on to the cap, and then you need to pull them in different directions. So that's the purpose of these things. We need two of them. They act like hands so that they can pull the sister chromatids apart in the end, and that's why they're moving to opposite sides of the cell. All right, so here's a picture of pro-metaphase. You can see that the nucleus is gone. These things, the chromosomes, they look like worms because they're condensed. We would never see these things. They would look like this if we were in interphase. But now we can see them. They're stained blue here. Here's our centrosomes, right? And remember, the centrochromatids are held together by centromeres. What does meros mean? Part, right? So what's a centromere?
middle part.
Yeah, exactly. So it's the middle part that holds these two sister chromatids together. These are sister chromatids. Remember, identical copies. It could be like a #1 from your mom. And there's two copies that were an extra copy was made in S phase. And that's held together by the centromere. So these centrosomes are building these microtubules, which are here in yellow, and their job is to attach to the sister chromatids on opposite sides. So this one's going to attach to this one, this one's going to attach to this one, and the object is to pull these things apart so that only one goes into each side of the cell. We call these things spindle fibers. These are the fibers that go across here. It kind of looks like a spindle. And they actually connect to a specific site called the kinetochore. So there are kinetochore microtubules, the ones that connect to the kinetochore. And then there are non-kinetochore microtubules. They push on each other to sort of stretch the cell out. So they're going to attach to one another, and there's motor proteins here, and it takes the cell and elongates it out to stretch it to get it ready to cut into two. All right, so metaphase is super easy. If you saw it under a microscope, you wouldn't recognize it. All of the sister chromatids line up in the middle of the cell. Some scientists named it the metaphase plate. It's not a real thing. It's just an invisible. It's just a pretend line. It's like the equator of the earth, right? There's not really a stripe down the middle of the earth. It's just what it's called. So the metaphase plate is the middle where all these sister chromatids line up. This whole thing collectively with the microtubules is called the spindle. It kind of looks like a hamster ball. Remember the sister chromatids are still attached to each other. The microtubules are attached to the kinetic cores. In anaphase, they separate. So the centromeres are like a molecular glue that hold these things together, and they gets dissolved, and then these things move. They have this characteristic V shape. Imagine if you took a piece of string and held it in the middle and then pulled it through a swimming pool. What shape would that piece of string have? It would make a V shape. So just try it. Put it in a bathtub or a cup of water, and grab the string in the middle and pull it. It's going to make a V shape because the cytoplasm is full of water and other stuff. So anaphase always has this characteristic V shape of these chromosomes. You can kind of see it here, and that's where they're separated. This is the microtubule organizing center. These green things are the microtubules, and then the blue ones are the chromosomes. So now they've separated into different sides, and so hopefully, if this one all went well, you would get 23 of your mom's chromosome on this side, 23 of your dad's on this side, so you get a total of 46 on one side, and then the same thing would happen on the other. And so we would have 46 chromosomes on this side, 46 on that side, and when we divide the cell into two, they would be two new genetically identical cells. All right, so telophase is basically the pinching of the cell into two. You can see that this thing, it forms what we call a cleavage furrow. The artist drew it here. Essentially, this is actin, same protein that moves your muscles. It actually acts like a piece of string tied around clay, and if you pull it tight, it will pinch the cell into two. And then you get two new daughter cells that hopefully are genetically identical. All right, so let me dig up a YouTube video of this whole process and see if you can identify this. And then you also have a lab on identifying these two. I think that this is a mitosis of a Drosophila, but I want to get one of I'm gonna see if I can find one of Xenopus. So Xenopus is an African clawed frog, and it's well studied because its eggs actually develop on its back. So you can see this process as it's happening. All right. So I'm going to stop sharing the PowerPoint and then we'll share this video. Okay, so I'm going to back this up. And so if I put this on the test, what phase would you say that this is in? Is it in prophase, pro-metaphase, metaphase? And
Is that prophase?
yeah, and the reason you can tell is because it has a definite round shape, like the nucleus is still present, right? But it looks like worms, so you know they're condensed. You can't really see the centrioles here because they're not stained, but they're there. And so let's just go on. And the next stage is pro-metaphase, and you'll see the nucleus stops taking on a shape because it completely comes apart so that those microtubules can access the chromosomes. So now you can see it doesn't have that definite nucleus shape, right? So this is pro-metaphase. And then what's the next phase? Do you remember?
Is it anaphase?
It's
Metaphase.
metaphase, yeah. And remember, metaphase, they all line up in that invisible plate. So there's a lot of pushing and pulling going on to line these things up, so that they're on this, invisible plane. It's not really well defined here, but you'll see it once it starts separating. But you can see that, okay, and so so right here is the metaphase plate. I mean, you can see that this is not easy to see, but in most cells it's more defined, especially in like plant cells. In the lab you'll see that they're all lined up in the middle. And then the next phase is anaphase, and that's when they separate. So they're going to do that now. And you can see the sister chromatids move to opposite sides. So that's anaphase. And then you can see that this cleavage furrow right here is starting to form with that actin to divide it into two new cells. Can you guys see that?
Yeah.
And so that would be what we call telophase. This is with it stained. So let's see if you can guess these. So what's this one?
Prophase.
Prophase, yeah, because the nucleus is still intact. The chromosomes look like worms. And then what phase is this?
Is it pro-metaphase?
Right, because you can't really see the middle yet, but it's starting to form. And then what phase is this?
Metaphase.
Metaphase, right? You can definitely see that line in the middle. And then oh, it's going to do a frog egg. So this is a xenopus egg. There's the metaphase plate right there. I can see that thing pretty clear. See it?
Yeah.
And then now it's lined up and it's going to do anaphase. It had actually it's kind of interesting. See this chromosome over here? So it's not lined up. That means it hasn't attached to the microtubule, and it will stop cell division until this thing gets in the middle. So that's kind of cool. That shows you how important this process is. It will stop. It will stop cell division in metaphase until that gets to where it's supposed to go. And now everything's there. It's going to divide anaphase, right? You're going to pull apart. And then we have the cleavage furrow and telophase and cytokinesis. All right. So any questions about that?
Nope.
I'm going to stop share this and we'll go back to PowerPoint. Okay. So the mitotic spindle is what drives the force of meiosis. The spindle pushes against each side, you know, kind of like we talked about with the pin, removing the cap and stuff like that. So all the forces are generated by these things, these microtubules here. Remember microtubules, you already learned this, they're made out of tubulin. You learned this when we talked about the cytoskeleton. And then you should know that these things are called centrosomes, and each of these little tiny parts of it are called centrioles. So just make some flashcards and learn that. The kinetochore is where the microtubule attaches. So we have our centrosome over here, and it's building this microtubule to attach to this sister chromatid here. And remember, it attaches at the kinetochore. So the artist has drawn this a little weird. It really doesn't look like that. It looks more like, so the kinetochore is actually a protein that's attaches to a sequence right on the outside, and then in the inside, this is the centromere. And this on the outside is the kinetochore. But they drew this for effect to show you that the chromosomes actually move themselves. And you can think about it this way. So let's say you're in a boat and you went skiing with some friends and you have your line out there and you're skiing, you're in the water. And so you're done, you want to get in the boat. So you could either pull yourself along the rope, right? Or someone could pull you in by the rope. Does that make sense? To get into the boat?
Yeah.
Okay. So how would you know the difference if like you were flying a drone above this boat? And the way you could tell is, let's put a dot here, right, on the rope. We'll just mark the rope. So if you pulled yourself in, would that dot move?
Yeah.
Now, if you pulled yourself in, would the dot move?
Oh, no.
Yeah, because you're pulling yourself along the rope, right? So the rope length doesn't ever change. Now, if someone pulled you in from the boat by pulling in the rope, then the rope would go in, right, to the boat, and so that dot would move. Does that make sense?
Yeah.
All right, so scientists didn't know if chromosomes pulled themselves in, kind of like a skier, or if it was the centrosome doing the pulling, right? Someone in the microtubule organizing center. And so they actually speculated that it was a centrosome, but then they did an experiment where they put a mark on these microtubules. And so once they marked them, they noticed that that mark never moved. And that means that the chromosomes are pulling themselves in, kind of like the skier, rather than someone in the boat pulling them in. Because this mark would move if it was pulling it in. Does that make sense? So that's how we know that chromosomes pull themselves in. So the artist that drew this is just showing you that the Lego pieces that make up tubulin are coming off the end, and that these little feet, remember all those little molecular motors that move your muscles and stuff, have these little feet, these motors. And so it's literally walking down the microtubule, which is kind of trippy, but that's how it works. Okay. Non-kinetochore microtubules, we talked about that. They push against each other and elongate the cell to prepare it to divide into two. No big deal. In animals, we had that cleavage furrow, which I showed you. It's pretty obvious. This is an electron microscope. What kind of electron microscope is this? Scanning or transmitting?
Scanning.
Yes, because we can only see the outside. So this is a cell that's divided, it's dividing into 4 new cells. This would be what we call a zygote, like the original fertilized egg. And plants, they have a cell wall, remember, so they can't do that. So let me show you a video of how plants do it. Usually this is a water lily because those are well studied. I might have to type in water lily. Give me 1 second. Okay. So I'm going to stop sharing the PowerPoint. And I'm going to share this video of this water lily, also known as a blood lily. Okay. So you can sort of play along with the phases of mitosis because they're the same with plants and animals. So see if you can figure out what phase this is.
Is that prometaphase?
We're in prophase because we still feel the nucleus. Now the nucleus is gone, so that's metaphase. Well, that's pretty fast. Yeah, it's kind of hard to tell. If you wrote prophase on the test, I would give you credit for that because you can't really see the well-defined nucleus. So prophase, prometaphase, and then a lot of pushing and pulling to get it in the middle. And that's the metaphase. It's kind of, you can kind of see it right here. And then anaphase. And then, so this is forming, this wall right here is formed from parts of the Golgi we talked about in chapter 6. And it is called a cell plate. It's eventually going to be a cell wall. Remember, cell walls are made out of cellulose. And so that's what makes plants different than animals, is because they have a cell wall. They they don't form a cleavage furrow. So make sure you know the difference between the two. There's not much. Animals have a cleavage furrow because they don't have a cell wall. Plants make a cell plate because they do have a cell wall. Remember that the ring, the contractile ring, is made from actin. You guys learned this in the cytoskeletal unit. Remember, microfilaments are made out of actin. And the motor protein that you already learned, you learned this in the cytoskeletal unit, is called myosin. So make sure you know that. It's just, that should just be review. And it just pinches it in two, kind of like we talked about dental floss around a piece of Play-Doh. And then we end up with 2 new daughter cells. So that's cytokinesis in animals. Plants, we just showed that. So we have the cell plate. It's made from parts that are from the Golgi. So on the test, if I ask you, what are the parts from the make the cell plate come from, you tell me the Golgi if you want to get it right. All right. And then that cell plate continues to grow until it forms a cell wall, which is the only difference in plants. So this is just the artist's rendition of it until it fully divides the cells into 2. So bacteria are kind of funky. They do something called binary fission. It's different than mitosis. Bacteria, so you guys know that we have chromosomes that are linear and they kind of look like cigars. And but bacteria have circular chromosomes rather than linear. So our DNA is a double helix. You know, theirs is too, but it's attached all the way through a circle. And they have a place called an origin of replication. We call it ORI, and that's where their DNA is copied. So that replication begins at a single point called the origin of replication. It's shown here. This doesn't really look like a circle, but it is, trust me. And remember, prokaryotes have cell walls, and they also have a plasma membrane. This is an E. coli. This is a Escherichia coli. This would be found in your intestines. Like right now, you have this in your colon, in your large intestines and colon. Anyway, nobody really knows how this works, but it is quite important because let's imagine if you could stop binary fission. If you figured out how this worked, you can figure out a way to stop it. What would happen? Let's say with MRSA, right? So it doesn't work very well with antibiotics. But what if you could stop binary fission? What would happen to the bacteria?
We'll kill it.
Die.
It wouldn't be able to replicate it, and it would die. So you could rid the earth of harmful bacteria by figuring out how binary fission works and stopping it, because it works different than our cells, so it wouldn't affect our cells. You know, the truth is that pharmaceutical companies aren't really interested in looking at this because they don't make a lot of money off antibiotics, because people take them and then they're done with them. You know what they make money off of is drugs that you have to take for the rest of your life, like lipid lowering drugs or high blood pressure drugs or things like that. And that's where they make their money. And so they don't even there's not a single pharmaceutical company working on any kind of antibiotic or any kind of research on binary fission to deal with this bacteria issue. But maybe they should. I'm just saying. All right. So this is what we do know, that bacteria have a circular chromosome. I might ask you on the test, what kind of chromosomes do bacteria have? Make sure you tell me they're circular. Those two, it copies itself, but it's still attached to the origin of replication. So there's two circle, another copy made. Those somehow get moved to the other end. We really don't know. So this origin gets folded over here. This one ends up over here. And then the membrane grows inward. Remember, they don't have cytoskeleton. So exactly how this works is not really well known. And then they end up with two daughter cells. So a lot of studies should be done on this. You know, I think it will be helpful in fighting bacterial infections in the future. But I'm just going to ask you a couple things about this, like, what's binary fission? Well, that's how bacteria do cell division. What do we know about it? Nothing. You need to know what an origin of replication is, because that's where it separates. And you need to know that it roughly doubles in size, just like we talked about before in regular cells. Okay, so if we look at this, then this will be the last slide we talk about today. So there's a big giant leap in evolution here. There are only 4 different ways to do cell division. We have the bacteria, which do their binary fission. We have most other eukaryotes that we talked about that do regular mitosis, right? The cell membrane, the nuclear membrane comes apart, the sister chromatids are pulled apart. Dinoflagellates and diatoms, and you guys need to know this because these are the 2 exceptions of all of the entire living systems that we know of that do mitosis differently. So it's pretty simple. Dinoflagellates, the nucleus stays intact and the microtubules attach to the nucleus itself on the outside and pull the nucleus apart and divide it in 2. Diatoms have those centrosomes inside the nucleus. And so all the nucleus stays intact and all this stuff goes on inside the nucleus. Whereas, in all other systems, the nucleus comes apart, right? And that gives access to it. So the nucleus stays intact. The nucleus itself is divided in two. And then this one, and the centrosomes are on the outside. This one, the centrosomes are inside the nucleus, and they attach and pull apart the citrochromatids. And then here the centrosomes are in the cytoplasm, and then the nuclear envelope comes apart. So on the test, I might ask you, give me some examples of organisms that do not do regular mitosis. And you would say diatoms, dinoflagellates, and prokaryotes. All right, so we're going to end here, and we'll pick this up on Tuesday. You guys have any questions?
Nope.
All right, I'm going to jump over to office hours, and then I'll post this on YouTube later on this afternoon.
Thank you.
All right, have a good one.